Good evening, everyone.
Good evening, sir.
Good evening, sir.
Okay, previous section we discussed about spontaneous and biogenesis theory, right? Previous day class. Any question from this previous session? Spontaneous versus biogenesis. At the end, we are disproving the spontaneous generation by the help of Louis Pasteur experiment, classic experiment, and he used a flask for this. So first controversy was done. First controversy was spontaneous or biogenesis. How the life was came. It was spontaneously or non spontaneously, and we are disproving that it is the life came from other living beings in case of microbes as well as other eukaryotic organisms. The second controversy was the germ theory of disease. That means the disease caused by microbes or another elements. One controversy gone, another came. The second controversy was germ theory of disease. What is disease in a single line? Human or any animals, plants health, affect health and uh, change the state from well-being to change the structure, state or function. So disease means change the body state, better body state and function. And we now know for every disease a particular organism involved. The statement is correct or false. It is the
Correct.
current theory. It is the current theory. We now know after very much research, we now know that for every disease, a particular microbes are involved. You can check any diseases and there is a particular groups. If you say about the pneumonia, there is a bacteria involved. If you say the current pandemic, COVID, there is a virus involved. So particular disease, particular organism cause particular disease. That is the current theory. But the previous time, ancient period, we know that at the ancient period, they does not knew about anything about the microbes. So they believe that in earlier state, earlier people believe that multiple theory about the disease theory, multiple fact about disease. They believe that number one, supernatural force, supernatural force cause disease. Supernatural force cause disease. Some believe that it's a God punishment. Disease is the God punishment. We can say about that in the two. Disease is punishment of God. Some are believed about poisonous vapor theory. Poisonous vapor cause disease. We have said out earlier multiple facts are there for the statement of disease. Supernatural force, poisonous vapor, God punishment. Number 4, imbalance among body four humors can also cause disease. Check this statement number 4. Fact number 4. That is four humors. Our body contains four humors, and the body humors when imbalanced does not maintain, then our body state will be changed, function will be changed, and disease appear. And the last statement, believed some physicians. These statements believed by some ancient physician. And one of the renowned physician at that time, the Greek physician Galen. Greek physician Galen. He believed in the theory number 4. That means changes among the four humors, body contains four humors, and changes the humors, any one of the humors lead to the disease. First three are crazy, but number 4 statements or fact has some meaningful way. And that's why some physicians are believe at that time, and the Galen, one of them, renowned scientist, renowned physician, that time believe or favor the fourth fact. That is the four humors cause diseases, imbalance. Now we need to know what are the four humors present in our body. The four humors are number 1, blood. Number 2, flame. Number 3, yellow bile. Yellow bile, another name colored. And number 4, black bile. Number 4, black bile. Another name, melancholy. These are the four humors contained in our body system. That means human body system, animal body system contain these four humors, and any of the humor imbalance cause or appear disease. So actually, they mention about blood means cardiovascular system. Yes or no? So actually, four humors maintain our different system. Blood maintain the cardiovascular system. So any changes in the cardiovascular system lead to different types of diseases. Cardiovascular disease, cardiovascular system. What is the meaning of flame? Flame means thick viscous substance. Thick viscous substance that release abnormally. from mouth and nose. So what they indicate by the means of phlegm? What they indicate, the thick viscous substance, we now know it is a mucus, and mucus is secreted from the mouth and nose abnormally. That means there is a problem in respiratory system. So indicate about phlegm. Humor number two, phlegm means respiratory system. Any problem in respiratory system, we know variety of diseases can appear after that. So phlegm means actually they indicate about respiratory system. Blood indicate about cardiovascular system. And number three humor is yellow wine. Another name is color. Yellow wine means greenish yellow liquid made and released by liver and stored in gallbladder. That is the yellow wine or color. So they actually indicate about the bile which is formed from the liver and stored in the gallbladder, and it helps in the digestive system. We know that the bile help in the digestive system, particularly lipid metabolism, lipid digestion helps the bile actually. So indicate about the third humor is the digestive system. Your digestive system. If any problem in the digestive system can lead variety of disease. Number four, another humor we present in our body structure is black bile. And black bile, another name is melancholy, produced during the stress condition, and it can be produced from kidney and spleen. This humor produced from kidney and spleen. Abnormally produced during the stress condition, and they indicate this humor or connected this humor with urogenital system, urinary and genital system, urinary system better. So at that time, the physicians, mainly Galen, connected our body structure with the four humors, and they said that the four humors, when in the balance condition, maintained in balance, there is no diseases. But any imbalances, changes lead to the diseases, and you can check this. Change in the blood composition, change in the any system problem, cardiovascular system lead to the diseases, as well as respiratory, digestive, and urinary system. So you cannot deny this statement. Can we? Can we deny this statement? We know that any change in the respiratory, cardiovascular, digestive cause diseases. But what cause changes? What cause changes in the cardiovascular system? What cause changes in the respiratory system? What cause changes in the digestive system? And that cause mainly caused by microorganisms or organisms. So we need to support this theory later, that germ theory of disease. So previously people believe many theory There is a logical theory in the fourth statement, but still we cannot cover all the things because changes occur. But what cause changes? They are not appropriately answered. So we now know that the changes caused in the blood, in the flame, in the yellow bile caused mainly by microorganisms and agents, other agents. So from that view, the statement is here, germ theory of diseases. What does it mean? Germ theory of diseases means a disease caused by germs. Germs mean microorganisms. Not all diseases, but majority of diseases. Not all diseases are caused by microbes, but majority of diseases are caused by germs, that is microorganism. So later, the statement given, that is germ theory of disease. Germ theory of disease. And it means germs, that is microorganisms, can invade other organisms and cause diseases and cause diseases. The meaning of this statement, germ theory of disease, means germs can invade, that means microbes can invade other body organisms, other body system and cause several diseases. Clear? So this theory has been supported by many scientists, by their work, by their research, and some scientists' contributions are number one is starting from Agostino Bassi. These scientists are favoring the germ theory of disease. Okay, supported the statement germ theory of disease. Number one, Agostino Bassi. In the year of 1835, he discovered that the disease called pebrine. The name of the disease is pebrine and affect silkworm caused by a fungal cell. Caused by a fungal cell. Pebrine disease affected the silkworm and caused by fungi. And we know that fungi is the microbes, microorganisms. So microorganisms can cause disease. First evidence come from Agostino Bassi work in 1835, pebrine disease. Number 2, M.J. Berkeley. (1845), 10 years later, Irish scientist. He discovered potato blight. Potato blight is a plant disease caused by mold. Caused by mold. Mold is a type of fungi, so it is also microbes cause diseases. And here in the case, plant disease also caused by microbes. Plant disease also caused by microbes. Here, silkworm, animal caused by microbes. So two evident come. Plant disease, animal disease caused by microbes. Augustino Bassi and M.J. Berkeley. Number 3, Henrik D. Berry. In 1853, he observed that variety of cereal diseases are caused by fungi. And these fungi he categorized into smart fungi and rust fungi based on disease appearance. So another consolidation come from the Dewey observation: fungi cause plant diseases, variety of fungi. And based on the disease appearance, he classified the fungi smart and rust. Clear? Number 4, scientist number 4. Any question? Clear? It's just a statement. Scientist number 4, Louis Pasteur. Louis Pasteur also contribute in this field also, germ theory of disease, but in indirect way, not directly, but indirectly. Pasteur also contribute in indirect way. How? Pasteur at that time theorized that the fermentation theory, statement about fermentation theory, the theory of fermentation. Yes, previous day class, fact paper class, fermentation, basics of fermentation. Who discovered? Louis Pasteur. Louis Pasteur discovered the fermentation theory. Fermentation theory. In fermentation theory, Louis Pasteur observed that or demonstrated that fermentation is caused by specific bacteria and yeast. Specific bacteria and yeast. Clearly, he mentioned the yeast and some bacteria change the material into alcoholic substances, change the glucose into alcoholic substances, and that is the theory of fermentation. By using this fermentation theory, at that time in France, wine industry are continuously grow by using this fermentation theory. Wine industry are rapidly surged because they know about the technique, know about the organism which change the glucose into the alcohol. So they start to produce production of wines. So wine industry, beverages industry, alcoholic beverages industry are rapidly surged at that time in the France. Louis Pasteur is French scientist, but that time the wine industry facing a new challenge. Wine industry facing that poor quality, poor quality of wine produced. The quality is not good, the taste is not good, the taste not appropriate. And call the Louis Pasteur that time and look into he look into this matter why the wine quality is poor or taste will change. Louis Pasteur, after clear observance, he found that certain microbes are involved. Certain microbes are involved to change the taste of the wine. And the poor quality wine means, here we write about the poor quality wine. That means wine become sour. Wine become sour. Taste will be changed to sourness. When Louis Pasteur he found the microbes are involved. Microbes are involved to change the taste. And during this inspection, he called this sour wine as disease wine. The sour wine called Louis Pasteur as disease wine. Pasteur found the remedy for this, and Pasteur clearly said that Heat treatment. Heat treatment can lead to eliminate undesired microbes. Eliminate undesired microbes. In his treatment was disease causing microbes. Actually, the undesired microbes nothing but acetic acid bacteria. What happened during the sourness? The ethanol, after production by yeast or bacteria that produce ethanol, ethanol converted to acetic acid. Ethanol converted to acetic acid by some undesired bacteria called acetic acid bacteria. They start to produce acetic acid and make the wine sour. And that is the main problem in the wine industry that time. And Pasteur found that acetic acid bacteria, the undesired microbes or disease-causing microbes in the wine, should be eliminated and the wine become fine. No sourness should be done. And this heat treatment procedure still we are using in variety of liquid treatment in as well as milk and called pasteurization. This heat treatment, special heat treatment to eliminate the undesired microbes are called pasteurization. Still are using rization to pasteurize the milk. Temperature change হবে. Temperature change হবে, but usually pasteurization temperature 72.5 degrees centigrade for 30 minutes, and then it will be varied. It can vary depending on the milk, depending on the liquid. So I said that Pasteur actually involved in this field by indirect way by means of fermentation theory, and he called the fermented quality of the wine when changed, poor quality, he called the disease wine. From this aspect, many scientists start to believe that microbial disease can be eliminated, microbial disease can be cured. Because he cured or he changed the undesired microbes, eliminate the undesired microbes by heat treatment. So many scientists believe that the microbes cause disease. That means deleted or eliminate those microbes, and the disease will not appear. So from this inspiration, next scientist number 5 came Joseph Lister. Joseph Lister. Number 5. Another indirect evidence came from English surgeon. He was a surgeon. Joseph Lister developed antiseptic surgery. Antiseptic surgery. Important statement. Question maybe come from that. Antiseptic surgery discovered by which scientist? Joseph Lister. Antiseptic surgery means there was a problem after surgery, variety of wound diseases will become and that could be life threatening. So Joseph Lister combat by using a phenol. So for antiseptic surgery, Joseph Lister used phenol. Phenol is a chemical disinfectant. Disinfectant means kill the disease causing organism. Phenol he used in diluted manner. Phenol, Joseph Lister used the phenol during dressing, surgical dressing, and sprayed over surgical area. By applying this phenol after the surgery, a remarkable achievement happened. That means he successfully reduced the surgical infection, rate of surgical infection. So what we can draw from this experiment, from this observation of Joseph Lister? What we can draw? We clearly state that the organism can be microorganism, the disease-causing organisms can be eliminated, and the infection will be reduced after that. And he inspired from Louis Pasteur fermentation. When Louis Pasteur fermented undesired microbes are killed by the means of pasteurization, that inspiration makes this happen. And he started to use phenol is a chemical disinfectant. And today also in we are using the phenol, Dettol, Savlon. These are also actually phenolic compound. These are also phenolic compound, not exact phenol derivative, phenolic compound. We are using regularly in our home, Dettol, Savlon, etc. So the phenolic compound has the beneficial effect to kill the microbes, first showed by the Joseph Lister. Clear? Joseph Lister, up to Joseph Lister. Any question?
No, sir.
Scientist number 6, Robert Koch. The final proof is came from Robert Koch study. Number 6. He confirmed the theory, germ theory of disease. Previous day class, the theory of spontaneous generation ended by Louis Pasteur. Here, the conformly eliminated the theory, conformly accept the theory of germ theory of disease, Robert Koch. He established a clear relationship between Bacillus anthracis. It is a bacteria. Bacillus anthracis and the disease anthrax. And the disease anthrax. First observed disease of Robert Koch, anthrax. Anthrax caused by cattle, caused in cattle, affected the cattle. Sheep are mainly affected. Sheep, cow, etc. And it is highly contagious disease. Anthrax can cause disease also in human, but rates are low, mainly affected the animals. How we prove that anthrax caused by the bacillus, that means a bacteria? He observed a few things. Observed and studied. A simple experiment. He discovered the thread-like organism. Thread-like organism. Why it is thread? Because the name of the genus is Bacillus. Bacillus shape is rod shape. It's a rod shape bacteria. So when he discovered the organism, it's a thread-like organism. In the blood of infected dead sheep. So he carefully studied the dead sheep that infected by the disease anthrax, and in their blood he found that the thread-like organisms. Clear? Then he took the blood and injected into healthy mice. What you will observe after these two steps, what happened? He take the blood from the infected mouse sheep and injected into the healthy mice in laboratory. What will happen? The mice will be died or mice will be affected by anthrax. So Koch also observed this thing, that is mice become dies. And another interesting fact observation is came from The dead mice blood observation. From the dead mice, the blood contain thread-like organism. Blood contain thread-like organism that he see earlier. Importantly, check the experiment. Clearly, he found thread-like organism in the sheep, dead sheep. And when it injected into the healthy mice, the mice become dies with the infection of anthrax. Then, after the careful investigation, he found the same thread organism. So, from this observation, can we tell these thread-like organisms is the actual infection or actual cause disease in the sheep? This thread-like organism actually causes the infection in the sheep? Yes
Yes.
or no? So
Yes.
Yes, sir.
from this observation, clearly Robert Koch stated that hence he proved that the disease anthrax caused by Bacillus anthracis. So this is a simple experiment done by Robert Koch during his time and proved that anthrax caused by particular bacteria, the thread-like organism, thread-like bacteria, Bacillus anthracis. Now we can determine from these simple steps that any disease can caused by any microbes, but a particular microbes we can uh, investigate by using these following steps. And these steps, the following steps are called Koch postulates. The following steps, these steps are called summarize the Robert Koch into Koch postulate. Important question. Koch postulates. What is Koch postulates? Actually, the steps to determine which organism cause which disease. So next topic, Koch postulates. Koch postulates help to identify the disease-causing organisms. It is some steps to identify the causative agent of disease. And Robert Koch summarized these steps, actually four steps, into Koch postulates. provide 4 steps in his postulates, and the 4 steps are: number 1, microorganisms must be present in disease organism but absent in healthy organism. This is it is the step 1, Koch postulate number 1, because during his observation of anthrax disease, he found that the dead sheep contain the thread-like organisms and the healthy sheep do not contain the threads. Yes or no? That's why he written this statement. The postulate number 1: the microbes must be present in disease organisms but absent in healthy organisms. Clear? So out of 4 steps,
Yes.
this is the step number 1: microbes present in disease organisms. And the step number 2, postulate number 2: the suspected organism must be isolated and grown in pure culture. Grown in pure culture. Pure culture means laboratory culture. Laboratory bacterial culture. Important postulate state: the organism which you suspect during the first inspection, the disease causing organisms, must be isolated from the disease organism, and then it should be grown inside the lab by pure culture method. Number 3, the same disease result when it inoculated in healthy host. So step number 1 done. After that, step number 2, grow the organism in the laboratory condition. After step number 2, you have to do step number 3. That means the organisms which you grown in the pure culture laboratory culturing method, this pure culture, when inoculated into the healthy host, you have to be choose a experimental host for that, and inoculated or injected this pure culture, and check same disease appear or not. Same disease appear after incubation or not. Rested for 3 or 4 days for incubation time. Give the time and check same disease appears or not. This is the step number 3 in postulate 3 of Koch postulate. If same disease appeared, then look for the step 4. Look for step number 4. The same microbes same microorganism must be isolated. from diseased host, from diseased host. The healthy host appear the same disease and take the sample from the disease and check the microbes. If the microbes is same in the previous, then we can call the microbes that we see in the previous actually cause the disease. So actually it's a guideline for causative agent determination. So first of all, microbes must be present in the disease host, absent in the healthy. Then from the disease host, the microbes are taken and grown inside the laboratory condition. The grown microbes have to be inoculated in the healthy host. For that, you have to be choose the experimental organism. Experimental organism can be guinea pig or mice. Inoculated that and check the same diseases appear or not. If same diseases appear, look for step number 4 from the disease mice or guinea pig. Take the sample and check the microbes. The microbes will be same as you see in earlier. If it is same, we can say the disease is caused by that microbes. Understood?
Yes, sir.
Yes, sir.
So for step number 1,
Yes, sir.
we for step number 1 for microbes, microorganism must be present or absent. For checking the microbes present in the disease organisms, we can use microscopy. So which tools are required for each step? Step number 1 need microscopy. Step number 1 need staining. What is staining, sir? Staining means to color the organism for better visualization of bacteria or others. Staining means to color the organism or bacteria to better visualization. We will discuss this topic in semester 2, staining. There are various stains are involved for bacterial staining purpose and what are their mechanism of action, etc., etc. Okay, so for step number 1, we need to use this tool, microscopy and staining, for observation of microbes present or not in the disease organism and absent in case of healthy organism. The tools used in the postulate 2 is microscopy and staining. The tools that we can use for step number 2, grown in the pure culture. So for that we use the tool bacterial cell culturing. Bacterial cell culturing technique. Bacterial cell culturing technique. Introductory class we said about the petri dish, solid media, liquid media, etc., etc., to grow the microbes, suspected microbes inside the laboratory condition. For that, we use the BOD to constant the temperature, maintain the temperature at particular level, particular oxygen concentration, etc. So, bacterial cell culturing tools are used during the step number 2, postulate number 2. Postulate number 3, inoculated into the healthy host and appear the same disease, we use experimental animals. For step number 3, the tools used experimental animals. Experimental animals we can choose guinea pig, and today all labs are used mice for experiment, various kind of experiment. Number 4, postulate number 4, we uses Lab re-isolation and culture. Lab re-isolation means same microscopy and staining, because we have to observe the same microbes, na? That is observed in step 1. So lab re-isolation technique means same, that is we use in the step number 1, microscopy and staining, and also the culturing technique, culturing tool. হ্যাঁ
স্টেনিং এ সাধারণত আমরা বিরোধীরাও এই ব্রঞ্জ ইউজ করতে পারি
স্টেনিং এ
সাধারণত বেল দিয়ে কি ব্রঞ্জ ইউজ করতে হয় কালার
একই তো, স্টেনিং মানেই তো কালার, কালারিং কম্পাউন্ড যেটা ব্যাকটেরিয়া সেল ওয়ালে ডিপোজিট হয়ে কালার প্রডিউস করে, দ্যাট ইজ দ্য স্টেনিং কনসেপ্ট। আছে এটা সেমিস্টার 2 তে ডিটেইলস, how staining work, what are the different types of stain we can use in bacterial culturing, bacterial staining, etc. Fungal staining also. So direct question can become, that is what are the 4 steps of Koch postulates, or indirectly the question can be given that what are the tools are used, what are the tools are used in each step of the Koch postulate. So it's a tricky question. Then 4 steps যদি লেখক তো নাম্বার হবে না, তোমাকে বলছি what are the tools are used, that means that tools are used in case number 1, postulate number 1, microscopy and staining are used, 2, 3, 4, etc. Different culturing tools and different tools are used during each step. So question can be vary from Koch postulate is an important topic for exam. Koch postulate. Clear? Koch postulate. Any question from Koch postulate? It's an important topic. Online, clear? Koch postulate.
Yes, sir.
So if it is clear, then answer the following question. Question number 1 from Koch postulate. Can you establish a link between tuberculosis and Mycobacterium tuberculosis bacteria? The causative organism of tuberculosis. How?
সর্বপ্রথম আমাদেরকে প্রথমে যেহেতু আমাদের যে কজিং এজেন্ট আছে
First of all, tuberculosis is the disease that cause in human. It's a human disease. And question wants the tuberculosis and Mycobacterium tuberculosis the link up.
প্রথমে আমাদেরকে দেখতে হবে যে টিউবারকুলোসিস রোগটা যে মানে মানুষের দেহে হয়েছে সেই দেহে মানুষের দেহ থেকে আমাদেরকে স্যাম্পলটা প্রথমত নিতে হবে।
So step 1, follow the Koch postulate. Follow the Koch postulates and write regarding tuberculosis and Mycobacterium tuberculosis. So direct statement লিখে দিলে আনসার হবে না। It's a tricky question. Direct লিখবো না, আমরা চেঞ্জ করব, মডুলেট করতে হবে রিগার্ডিং tuberculosis and Mycobacterium tuberculosis. Answer me. How it will be modulated? How the Koch postulate would be modulated?
সর্বপ্রথমে স্যার আমরা প্রথমে টিউবারকুলোসিসের এজেন্টটা আক্রান্ত মানুষের থেকে আমরা নেব।
First check the microbes, that means Mycobacterium tuberculosis. Check Mycobacterium tuberculosis in diseased patient and healthy person. For presence or not. First of all, it will be identified prior. Go to the check step number 2. That is tuberculosis. The Mycobacterium tuberculosis present in the diseased person, diseased patient. If answer is yes, present, and in healthy persons, it will be absent. The answer will be after statement 1. In diseased person, Mycobacterium tuberculosis present. Answer diseased person yes, and the healthy person absent. So step number 1, guideline number 1 match. Go for guideline number 2. What will be modulation?
যেটা থাকবে সেটাকে আমাদের আলাদা কালচারাল টিউব বার করাতে হবে।
So culturing Mycobacterium tuberculosis in particular media in laboratory condition. So second হবে culture M tuberculosis in laboratory condition. And for this, we use coagulated blood serum. Coagulated blood serum. Otherwise, M tuberculosis could not grow. So growing the microbes, growing the Mycobacterium tuberculosis in coagulated blood serum in laboratory condition and make the pure culture. Statement number 2 is done. Step number 2 is done. Culturing done. After that, healthy person. Voluntary কে হবে healthy person?
Mice.
Person তো হবে না, animal, obviously. Number 3, after that growing into the laboratory condition, the pure culture is taken and injected into the experimental organism, not in the human. Human কেউ voluntary করবে না, কেউ tuberculosis হবে চাইবে না. So number 3, step number 3, take the pure culture of M tuberculosis and injected in guinea pig. Better take guinea pig here. Also, we can establish mice disease in the mice also. And look for same disease appeared or not. This M tuberculosis produce the same type of symptom in the mice or not. Check for symptoms of tuberculosis, of disease tuberculosis. What will be the answer? The answer is yes, same type of disease symptoms are appeared in the healthy mice. Same type of symptoms. After that, the statement number 3, step number 3 also validate. Go for step number 4. The affected mice or guinea pig. Affected mice that show the symptom of tuberculosis contain This M tuberculosis or not, further you have to be established. Affected mice contain M tuberculosis bacteria or not, have to be checked. If the answer is yes, the affected mice, healthy mice in laboratory condition produce same Mycobacterium tuberculosis, answer is yes, then we can say there is a link between Mycobacterium tuberculosis and tuberculosis disease caused in human. Yes or no?
Yes.
If the four steps are validated, then we can set a link between tuberculosis and Mycobacterium tuberculosis bacteria. Hi Anna.
Yes.
Yes, ma'am.
So for any causative disease isolation, we need to validate the four steps of Koch postulate. Clear?
স্যার
Huh.
প্রত্যেকটা রোগের জন্য তো আপনার এই সেল মানে কালচারিং করার জিনিসটা আলাদা যেমন এটার জন্য কোয়ালিফাইড ব্লাড সেল এবার প্রত্যেকটা রোগের জন্য কি আলাদা মনে রাখতে হবে
না মনে রাখার তো এত দরকার নেই, তোমাকে যে কোনো একটা কি দুটো দেবে, এইটা তো দেবে, আর একটা যেটা কজ করেছিল ইন ল্যাটার, সেটা আমি কোশ্চেন নাম্বার 2 এটাও কজ করেছিল, এস্টাবলিশ করেছিল। তো কজের ফার্স্ট অবজারভেশন ছিল অ্যানথ্রাক্স disease establishment, after that tuberculosis disease establishment. This is also done by Koch, Robert Koch. And question number 2, it is in the homework HW, and submit this homework in WhatsApp after written individually. Another disease establishment. Another disease establishment. That is the same question. Pneumonia and Streptococcus pneumoniae. Marks will be 3 or 4. So answer like this, 3 or 4. I give you hints that Streptococcus pneumonia for growth need blood agar. Pneumonia, Streptococcus pneumonia need blood agar for growth. So mention where it is required, blood agar. Okay. And second hints, Streptococcus pneumonia grow better in mice. Grow better in mice. Give with experiment also done by S pneumonia, but is different aspect. The DNA isolation, transformation. So it's a homework for next day. আর যেটা কোশ্চেন করছে ডিফারেন্ট অর্গানিজম ডিফারেন্ট গ্রো করবে, হিন্টস দিয়ে দেবে, it's provided, it will be provided in the question, where it is grow and in which experimental organism you are taken during the experiment. So in case of S pneumoniae it is mice, and S pneumoniae grow better in the blood agar, so that is the hints. and you have to be answered by following Koch postulate. So done for this day. Topics ends. Koch postulate ultimately give the experimental proof that germ theory of disease, that means organisms caused diseases, microbes cause diseases. Clear? Next day we studied about the extension of Koch postulate, modification. Nowadays, Koch postulate is modified and modulated versions are used to determine the link between the causative organism and the disease. So modification of Koch postulate, next day topic. So modulation is done. That means there are some limitation in Koch postulates. So we have to know about the limitation, modification, and why it will be modified. So some limitation of Koch postulate, etc. So Koch postulate topic, important topic. Next day also it is important. What is the limitation? What are the limitation of Koch postulate and how will you overcome the limitation? That means modification of Koch postulates. Now we are use molecular Koch postulates. Molecular level মানে molecular biology, we are helping molecular biology to determine the disease causing organisms and disease establishment theory. So it will be modified. How it will be modified, who modified, and what are the steps in molecular Koch postulates, discuss in next class.